Okay, well, hey, good afternoon, everyone. It's 11 o'clock, or it's 11 o'clock my time, noon Central Time. And since that's the time we've scheduled the webinar for, we'll go ahead and just kind of get started with things. So let me just kind of take a second and introduce myself and talk a little bit about this webinar today. So many of you guys may know me. My name is Dr. Steve Friedenberg. I am a criticalist and veterinarian at the University of Minnesota. I am also a geneticist, and I'm kind of delighted to be here today to talk to all of you guys about the research that we've been working on over the past 3 or 4 years to study the genetics of Addison's disease in Portuguese water dogs. I've actually studied this disease in standard poodles for many years and was kind of excited when an opportunity came up to research this disease in Portuguese water dogs as well. I will sort of say in terms of this whole webinar and the information we're going to talk about today that I think this is just the beginning of the journey in trying to kind of understand what's going on. But as the tools we have as geneticists, is our ability to kind of think about some of these inherited diseases improves, I think we'll kind of continue to make progress in the right direction. So before I get into the specifics of the slides and the information, let me just give some kind of details or logistics for the meeting so that everyone knows just how to think about what's going on today. So first of all, this meeting is being recorded. I think Janet will post the contents of the meeting to various Facebook pages and websites that you guys run. So for folks who were not able to attend the meeting today, you'll be able to kind of watch it at a later date. A couple other just kind of ground rules that would be really helpful since we're doing this on Zoom. I set the settings so that when you enter the meeting, everyone is automatically muted. And so if you could please just leave your microphone on mute while I'm talking through the first 40 or so minutes of the presentation, just to minimize background noise for the sake of other participants who are listening, that would be fantastic. And I think this whole presentation will probably take, I would say, maybe 40 minutes or so, could be a little bit more, could be a little bit less. But at the end, once I'm done with the presentation, there'll be an opportunity for everyone to kind of unmute themselves and ask questions either via the Zoom or via the chat. I would just ask that when you guys do get to the point of asking questions, that people raise their hand and then I can call on people so it's not like a free for all to answer questions and people can just aren't talking over each other. And I think that's kind of it as far as the logistics of the talk is concerned. And hopefully you can all hear me. Can you guys just all nod your head to make sure you're hearing this all okay, and to make for the few of you that I can see, can you all hear me okay? And everything that I'm saying? Yes. Yes. Okay. I think I see a few people nodding their head. Okay, great. That's awesome. Okay. So let's go ahead with the talk. And I'm going to first start off with an outline of the presentation so that you guys know what to expect throughout the talk. So first, I'm going to go through a review of what Addison's disease is, just so we're all on the same page about what we're talking about here. And then I'll review the study goals, so how we approach thinking about the study. Then I'm going to spend a while explaining the methods that we used to try to sort through what's going on. And this is really a summary of how we think about gene mapping in today's day and age, which is very, very bioinformatics focused. And then we'll talk about what are our findings been to date regarding the genetics of Addison's disease, and then we'll kind of move on to next steps. I think as I mentioned earlier, this is the beginning of the process. It's not necessarily the end, and so we definitely have more things that we'd like to continue pursuing. Okay. So let's start off and first talk about Addison's disease. So Addison's disease, as many of you guys who are listening to this talk know, is a disorder of the adrenal glands, right? And the adrenal glands are these organs that live, I'm just gonna make sure that you guys can see here. Here's like a laser pointer. These organs that live just on top of the kidneys, and the adrenal gland and if you're a dog, we kind of rotate everything 90 degrees, so they live just in front of the kidneys. And the left adrenal gland actually looks a little bit different from the right adrenal gland when we sort of look at it anatomically, but functionally they're the same. When we look at the adrenal glands, a lot of times when we visualize them in dogs, we look at them under ultrasound. And this is a picture of the right adrenal gland in a dog. And you can kind of tell because it looks a little bit like a peanut. It's this black thing in the middle of the screen. And one of the interesting things that you notice when you even look at this adrenal gland is you can sort of maybe tell that the adrenal gland has a little bit of an interesting architecture to it. So if I were to annotate the screen, I'm going to kind of try to choose the zoom pointers. There's kind of this part of the adrenal gland, and then there's another part of the adrenal gland kind of on the inside. And so this sort of speaks to the fact that the adrenal gland can have both a cortex and a medulla. And so there's two layers of the adrenal gland, and it's sort of a very, very complex organ. And so just delving into this, because I think it helps us understand what's going on with Addison's disease a little bit more. Actually, let me think I need to get rid of this before I can advance my slide. No, I can't seem to advance my slide here. What's going on? Hold slide for a second. How can I get back here? Oh, here we go. Okay, great. So this is what the adrenal gland looks like if you were to kind of look at it under a microscope. So it's kind of composed of these three layers, this outer layer called the capsule, then this next layer, which is called the adrenal cortex, and the inner layer, which is called the adrenal medulla. And so that sort of just speaks to its overall complexity. And when we think about the capsule is really just something that keeps the gland in place. Functionally, we sort of break the adrenal gland up into the cortex and the medulla. And the medulla is actually a totally and completely separate organ. We think of it as more as part of the nervous system. And we all learn in sort of basic biology about like the fight or flight response. And the fight or flight response is primarily controlled by a hormone called norepinephrine or adrenaline, and that's released from the adrenal medulla. And interestingly, in Addison's disease is a disease only of the adrenal cortex. So the adrenal cortex, essentially in dogs and people with Addison's disease, becomes destroyed, and the dogs and the people can no longer make the hormones that are really necessary for life, whereas the entire adrenal medulla is completely spared. Okay. And thinking about the cortex, if we were to sort of think of this and like double click on the adrenal cortex to kind of zoom in a little bit more, the cortex itself is actually a pretty complicated organ. And let me sort of explain this to you here. So you can see the picture on the left is a little bit more of a cartoon diagram where you have the inside of the medulla and the cortex and the capsule. And here, as you move toward the middle of the diagram where you're looking at it under a microscope, you can see some layers of the adrenal cortex. And pathologists break this up into three layers. One is called the zona glomerulosa, the next layer is called the zona fasciculata, and then the inner layer is called the zona reticularis. And in dogs, this zona reticularis is actually pretty poorly developed. So the most clinically important zones of the adrenal cortex are the glomerulosa and the fasciculata layer. And why is it a little bit important to understand what's going on here is because all of these layers secrete different hormones that are important for life. So the zona glomerulosa secretes these hormones that are called mineralocorticoids. And the most important of those hormones is a hormone called aldosterone. And one of the things that aldosterone does is it helps regulate salt and water balance in the body. And many of you guys who know some things about Addison's disease know that dogs with Addison's disease, for example, develop very high potassium levels and very low sodium levels. And that's all mediated by the lack of aldosterone that they have. This other layer is the zona fasciculata, which secretes hormones called glucocorticoids. The most important one in the dog is called cortisol. And cortisol is essentially our stress hormone, right? So it's released whenever the body is in a stressful situation, and in a variety of ways helps the body cope with those stressful situations. So if we think about it, what happens in dogs with Addison's disease is for some unknown reason, right? And we sort of have long suspected that this is an immune-mediated disease in dogs like it is in people. If we were to look at a dog with Addison's disease, this is actually a picture of the adrenal cortex or part of the adrenal cortex of a dog with Addison's disease. You can see this kind of cluster that I'm highlighting here with my marker. I'll see if I can just point this out with a drawing here, if I can get back to this thing. So right here that I'm highlighting in green, I guess that's really hard to see. 1 second, let's see if we can get a thicker line. This area that I'm highlighting in green, this is a whole cluster of inflammatory cells that have started to invade the adrenal gland of a dog with Addison's disease. And these inflammatory cells are essentially destroying the adrenal cortex, the cortical tissue, and making it impossible for the cells that would normally secrete hormones to actually do their job and secrete those hormones. So just kind of moving on. So what do we actually see clinically without these hormones? Is we start to see many of the effects that we recognize in patients with Addison's disease. So we start to see low sodium because we lack aldosterone that is normally mediated by that outer layer, the superior layer, the outer layer of the adrenal cortex. We see high potassium because the kidneys can't get rid of it. And then we also start to see low blood sugar in patients with Addison's disease because cortisol is one of these hormones that helps the liver produce extra blood sugar, and especially in stressful situations. And without cortisol, we see low blood sugar in our patients. We also see our patients can develop shock and dehydration from excess water loss that's mediated by the loss of aldosterone. Their bloodstream can start to get very, very acidic, secondary to the shock, and animals have difficulty maintaining the stability or the integrity of their blood vessels. And these different effects that we see in patients with Addison's disease, I've just highlighted here in purple, the sodium, potassium, shock, dehydration, and the acidosis, the low blood pH, are all mediated by aldosterone. And these effects, the low blood sugar and the difficulty in maintaining the blood stability, those are all mediated by cortisol. So that's kind of what we see in patients. And many of you guys know this is a treatable disease. We can replace those hormones. This is not a talk about treating Addison's disease, so we're going to sort of not focus on that for the remainder of the time that we have here. But that's essentially what we see clinically. Now, I want to sort of transition and think a little bit about the epidemiology of Addison's disease because it's relevant to some of the things that we're going to talk about in genetics. And many of you guys have seen this slide before, but I want to sort of make you guys all aware of it if you have not seen it before. So, in veterinary medicine, one of the things that can be difficult is that we really don't have many good epidemiology studies about the prevalence of diseases across breeds. And fortunately for us in thinking about studying Addison's disease is actually an exception to that study. There was a really nice study that came out of Sweden about 10 years ago at this point. But one of the interesting things about dogs in Nordic countries is that many of them, or way more of them than in the United States, have health insurance or pet insurance. And these investigators were able to go through the database, the health insurance database of over 500,000 dogs in Sweden. And what they used this information for was to try to understand what is the prevalence of Addison's disease across breeds. And so you have to take this with a little bit of a grain of salt, right? We're here in the United States. Some of you guys might be from overseas, but I presume most of you guys are from the United States. This represents a Swedish population of dogs. So is it exactly representative of an American population of dogs? We don't know, but clinically, this certainly resonates with what we see in the clinic here. And just to kind of explain this diagram here. So on the left, you have not all of the dogs they included in the study, but the top, some of the breeds that they included in the study. This next column, IR, is the incidence rates of Addison's disease, and that's measured in something of 10,000 dog year adjusted rates of years. Kind of not super relevant, but what you sort of see here in this column here on the right where it's labeled RR, that stands for the relative risk. So what does that mean? That means what is the risk of a particular dog breed developing Addison's disease compared to an average dog in this study? And what you notice in looking at this chart, this figure here, is that the two top breeds that are highly overrepresented in the Swedish population are Portuguese water dogs and standard poodles. And to sort of repeat what this figure says, this means that within this population of 500 plus thousand Swedish dogs, Portuguese water dogs were 29 times more likely to develop Addison's disease than an average dog. And standard poodles were 17 times more likely to develop Addison's disease than an average dog. And there's something called a p-value, which represents statistical significance. And both of these values were very highly statistically significant. There are other breeds that are overrepresented for Addison's disease in the study: bearded collies, Cairn terriers, Great Danes, but far and away, Portuguese water dogs and standard poodles were the most highly overrepresented breeds. And from a geneticist perspective, this is actually a really, really interesting thing, particularly when you look at this next figure over here. So I realize this is a little bit hard to read, but I'll sort of point out the relevant bits of it in just a second. So this is what's called a cladogram. We sort of call this the dog breed wheel, colloquially, when we're talking about it. And it comes from a paper by Heidi Parker, who works in Elaine Ostrander's lab. She's a very, very well-known geneticist. And what they do is they sort of use genetic markers to identify how do breeds group together and how closely related breeds are together. And from the purpose, and we're sort of not going to delve into all the kind of interesting nuances of this figure, but I want to point out this sort of what is most relevant to this talk. If you guys can see this circle, this oval toward the top of the page, what I've done is I've highlighted the two breeds in question here that have a high incidence of Addison's disease, which are standard poodles and Portuguese water dogs. And one of the things that you can see is standard poodles and Portuguese water dogs are basically the nearest cousins to each other. So what does this mean to us from a genetic perspective? It means that, well, these breeds are highly related. They're both highly overrepresented for Addison's disease. So we would hypothesize that Portuguese water dogs and standard poodles share at least some common risk alleles or risk genes or risk variants that predispose them both to developing Addison's disease. That would at least be a reasonable genetic hypothesis. Is it necessarily the case? No, but it certainly gives us a little bit of ground and approach to thinking about this disease. So kind of with all that information kind of taken together, I want to sort of move on and talk a little bit about the study now that we know a little bit about Addison's disease and now that we know a little bit about the incidence of it, as well as the breeding commonalities between the two highest at-risk breeds. So the goals of this study, which is funded by the Portuguese Water Dog Foundation through a grant done via the AKC, which is to identify the genetic risk factors for Addison's disease in Portuguese water dogs using cutting-edge genetic techniques. So there had been prior to this study, there had been a couple of papers in the literature studying this. They were dated back a really long time ago using kind of older marker sets that we have sort of much better techniques and approaches that we can use these days. And we really wanted to focus on two main things. So one is called the major histocompatibility complex genes. So these are genes that are really important in the immune system. Because what they do is they essentially scan the environment for things that represent self, meaning things that we shouldn't develop an immune reaction against, versus foreign things like bacteria, viruses, fungi, things that we should develop an immune system reaction against. And so these genes are called MHC, or major histocompatibility complex genes. As a geneticist, we need to study them separately because it's there's so much complexity in them. We need to sort of focus on those genes specifically when we study them. I'll give you some more details on that in a second. And then the second thing would be to do something called a genome-wide association study. And we'll talk about what that is as I work through the slides here. But this is essentially a way that we use these days to map genes that are associated with traits. And the second goal of this study was to sort of try to identify risk alleles or risk genes or variants that are shared between Portuguese water dogs and standard poodles using data that I've collected over the years and from ongoing work that's funded by the Poodle Club, the Poodle Club of America Foundation, to try to bring what we know about these two diseases, these two breeds, and the same disease and the two breeds together because a lot of times when you're sort of trying to do the same thing in two breeds, you have more power to figure out what's going on here. And so I want to sort of explain to you guys how we think about mapping genetic diseases. And to do that, I'm going to sort of explain a little bit about dogs as a genetic model and why we think how we sort of approach this information. So, and this is stuff that you guys, as breeders or at least Portuguese water dog fanciers are probably really familiar with. So you all know that dogs and wolves sort of originated from a common ancestor. And we think that maybe 15 to 30,000 years ago, dogs started to become domesticated. And there were these really ancient bottlenecks that have separated dogs from wolves. Now, what's really important from understanding breed-specific diseases is to understand that breeds, right, have developed over a much more recent time period. And this is going to differ for every breed, but we think of recent bottlenecks that led to the formation of breeds as having been 200, 300, 400 years ago. So not very long time ago. And in the formation of breeds, all breeds underwent sort of these bottlenecks where a small founding population led to a much larger population of dogs. And what happens genetically when you have these recent bottlenecks is that breeders selected, and this sort of goes for all breeds, applied human or artificial selection to make breeds look and behave in a certain way. And all of that limits genetic diversity within a breed. We obviously, as breeders, try to maximize genetic diversity, but it always is in the context with trying to maintain a breed standard. And this leads to certain diseases becoming overrepresented within a breed, right? And so this is very, very common. You sort of can think of for example, cardiac diseases. Some are common in Doberman Pinschers, and some are common in Great Danes, and some are common in little breed dogs, the same thing. And so this all relates to these recent bottlenecks that led to the formation of a breed. Now, what does this mean, right, in terms of genetic diseases that are going on? And so this is something that I sort of like to think about because I think it relates a bit to the breeding history, but also relates to what we're going to talk about and what we're going to find when I describe the findings for the Addison's disease study. So the first kind of disease that we can see are called Mendelian diseases, right? And so what Mendelian diseases are is they're diseases where you essentially have one mutation causing one disease. And many of these diseases have been described in dogs. There's at least, 500 plus diseases that are caused by these single point mutations. And what this means is, for example, many of you know that DNA is composed by like A's, T's, G's, and C's. You can have an A gets mutated to a C, and all of a sudden the presence of that C can lead to a change in a protein, and that can cause a specific disease. So that one change causes that one disease. For the most part, the reason why we see these diseases is when you have, for example, a popular sire, right? So you could have a sire that developed some disease. That sire's semen gets used to sort of breed many dogs throughout a breed, and the prevalence of that allele becomes more common and can potentially lead to this Mendelian diseases. The second kind of diseases that we can kind of see across within the dog breeding community are called polygenic diseases. So with polygenic diseases, this is where you have more than one gene. You could have 2, 3, 4, 5 genes that are needed to kind of come together in just the right combination to cause the disease. And so these are essentially, we think of common alleles or common variants that just happen to occur within uncommon combinations and lead to the formation of disease. And these kinds of polygenic diseases are much harder to map, because we need a lot more dogs to include and to try to sort through what's going on here. But there's a couple of examples in the veterinary literature where people have started to do this, but it's really not something that either in the human breed genetics community or the dog genetics community, we've done a great job in explaining this, but we're getting there as our ability to kind of, as our ability improves to sort of map genes. And then the final thing, which I think is an important thing to understand about dog breeds, dog diseases within dogs, is that to some extent, we sort of come to realize more and more that many breeds may have diseases that are essentially fixed within the breed or highly fixed within the disease. So essentially what this means is that like all dogs within a breed could be predisposed to the disease, right? So, and then what really determines whether a dog is going to get the disease could be whether or not a dog is exposed to a particular, for example, environmental antigen, right? So, and so these diseases become much, much harder to map because essentially all of the dogs look the same from a genetic perspective, but you need to start to think about what they're exposed to. But we can map these diseases if we start to look at what are the differences between breeds, right? So I'll just kind of give you an example to sort of explain this. Let's say we take a breed like a Portuguese water dog that's highly predisposed to Addison's disease. Well, if we were to compare genetically the Portuguese water dog to like a greyhound, which has a very, very low incidence of Addison's disease, maybe we'd be able to map the trait that causes Addison's disease because all Portuguese water dogs would have it and no greyhounds would. Now, you could imagine that's a really, really complicated thing to do because there's a lot of differences between Portuguese water dogs and greyhounds, right? Portuguese water dogs have curly or wavy hair and greyhounds have straight hair. They look completely different, right? There's going to be a ton of genetic differences between Portuguese water dogs and greyhounds, but among those differences could be the predisposition to Addison's disease. So it's something we can do, but it's a lot more challenging to do these crossbreed studies. Okay, so let's focus on the study that we performed, and I'm going to go through the methods for you guys, and then we'll talk a little bit about the results. So for this study, this is just sort of a filter or kind of a funnel to sort of show you guys how many samples we collected. So for the study, we collected a total of, and this really is thanks to you guys in the Portuguese water dog community, as well as, us going to shows and sending emails and Facebook groups and all sorts of things, as well as a collaboration that we put in place with Dr. Anita Oberbauer, who's also a dog geneticist at UC Davis. And so as part of this study, we collected a total of 471 DNA samples. 190 of them came from Dr. Oberbauer and 281. And this is an ongoing process, by the way. So these numbers, are give or take correct at this point. We collected an additional 281 dogs as part of the study. These dogs mostly come from the US and Canada, but to some extent they come throughout the world. They are from throughout the world. And sort of as we think about the filter, right, down from these 471 dogs, I want to sort of point out that the next sort of level of filtering that we do on these dogs is to sort of prune them for relatedness. And so when we prune these Portuguese water dogs, we sort of look at their pedigree, and then we also have genetic tools to try to evaluate how related they are. We go from almost 500 dogs to nearly 100 dogs. So that's sort of like a five-fold difference in terms of what we have in our bank versus dogs we can use for a gene mapping study. And the reason why it's such a severe decline in the number of dogs that we can have is because if we were to include dogs that are highly related to each other, it can quickly confound or confuse our ability to sort of map genes, because we start mapping things like what is this population that's within the Portuguese water dog community that's highly related. You start mapping just differences between another Portuguese water dog, like differences within the community that don't necessarily speak to what's different between dogs with Addison's and without Addison. So it sort of easily skews our results, and we need to drastically minimize the relatedness in our population. And so after sort of pruning these 114 down to 114 dogs, after we sort of get to other factors that are important, like things we want to balance, like sex, color, coat type, because all these things you really want to balance when you do a genome-wide association study, we sort of boiled this down to 46 affected dogs and 55 healthy dogs, right? So that was sort of the population that we are going to talk about when we get through some of the results. So this is really the first thing, is just collecting samples. Okay, so once we collect these samples, we extract DNA, and sort of what do we do with them? So one of the first things which I mentioned we were going to do is we were going to do this thing called MHC genotyping. So in animals and dogs and people, there's generally two classes of MHCs. They're called class 1 and class 2. This figure just comes from humans, so I'm Xing this out. In dogs, we have this gene, this MHC class 1 gene. It's called DLA88. That stands for dog leukocyte antigen 88. And DLA88 is probably the most important, but not the only class 1 gene. And then class 2 genes are commonly associated with autoimmune diseases. They have these other funky names. They're called DLA, DRB, DQA. That should say, the next one should say DQB, DRB, DQA, and DQB. Sorry for repeating that twice. So those are the names of the genes that we genotyped using a technique called Sanger sequencing. And then we sort of figure out what's going on with each dog in our population. And we look for, are any of these DLA genes associated with Addison's disease? Okay, and we'll talk about the results of that in just a second. So the second thing which we did for this study, as I mentioned, we were trying to do something called a genome-wide association study. And I'm going to explain to you now how we do that genome-wide association study. So one of the things that we wanted to make sure we did for this study was to apply the most cutting-edge genetic techniques to understanding Addison's and Portuguese water dogs, because we thought it would be the best likelihood of us finding anything. And sorry, my screen's not advancing here. Okay. So the way we actually do this genome-wide association study is we have to start out getting our genotypes, which means dogs like humans have about 3 billion bases in our genome. And we wanted to understand what's going on at nearly every one of those 3 billion bases in the genome. And the way we start doing this is we actually use a computational technique called imputation. And we did whole genome sequencing at a very, very high coverage of about 31 Portuguese water dogs, where we sort of, for all of these Portuguese water dogs, we knew every single genotype at all 3 billion bases in their genome. And the next thing we do is we use a technique called low-pass whole genome sequencing, where what you do is you sort of use, it's a much cheaper method to get these genotypes, but you can see you only get a couple of them because you're sequencing them at much, much lower depth. So for example, in this dog, right, we only know 1, 2, 3, 4, 5, 6 genotypes, whereas there's, maybe 50 genotypes available. And in Portuguese water dogs, we did this for all 101 dogs in our population. And then what we do is we use this genetic computational technique called imputation to actually guess the genotypes. And it's more than just, it's a very, 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 very educated guess. Think of it this way. And to sort of fill in all of these blanks. And so now essentially we have 101 Portuguese water dogs that we have millions and millions of genotypes that we've achieved through this computational technique at a really, really cost-effective method. And this is essentially how we do genome-wide association studies these days, is by getting these genotypes from imputation of low coverage data. And so that was the approach that we took here. So once we have all of these genotypes, what do we do with them? How does a genome-wide association study work? So essentially what you do is you take all of the genotypes in the genome, and this is just sort of an example of one strand of DNA, where you're looking at about 10 or 12 genotypes, TGA, GA, GTCC. And you sort of scan all of these million genotypes at once in all of these 101 Portuguese water dogs, and you try to ask yourself the question, in locations where there's variation in the genome, so here's an example where you could have a G or a C, an A or a G or a C or a T, we call those single nucleotide polymorphisms. And you scan the genome and you try to find one where, for example, in this G, all of the dogs are healthy, and all of the dogs here that have a C are unhealthy. And if you find a genotype like this that replicates across the genome, you've found a marker where all of the dogs that have the disease have one marker, and all of the dogs that don't have the disease have a different marker. And then we can sort of focus in on those regions of the genome that we think are associated with a particular disease. So that's how a genome-wide association study works. On top of this, we employed a parallel approach in standard poodles, where we had 463 dogs that we collected. We did MHC genotyping on 158 of them, and we actually did a smaller number that 93 of them were genotyped by low-pass sequencing. We used a bit more of a stringent relatedness cutoff than Portuguese water dogs, just for a whole host of reasons, but we really undertook a parallel approach in standard poodles. And the reason for that is so that we can try to say, okay, what do we find in Portuguese water dogs? How does it compare to the most closely related breed? Okay. On top of this, this is the last bit of the methods. I want you guys to know that, and many of you guys may have participated in a study, I mentioned that for complex diseases where we think there's more than just the net genetics at play, we undertook an environmental risk factor study where we surveyed standard poodles and Portuguese water dogs across the US and Canada. We enrolled a total of about 350 plus cases and over 4,000 controls. Asked people a broad range of questions, and we overlaid that data with information from the Environmental Protection Agency on like soil, air quality, water quality, et cetera. And the purpose of this was to try to say, can we combine the genetic data with environmental data to try to sort through what's going on here with Addison disease? Okay, so that was the overall approach. So let's now start to take a look at some of the results that we found. So the first thing which I mentioned is we did this study looking at MHC genes. These are the major histocompatibility complex genes, to try to ask ourselves, is there an association with Addison's disease? And we actually did find something really interesting here. So I'm going to walk you through this figure. So this is a picture of a Portuguese water dog. This pie chart on the left, which is kind of in the middle of the figure, is for cases. And this pie chart on the right is for controls. And each of these different colors, the red color, the gray color, the green color, the blue color, represents a different set of MHC genes. And we group them together into something we call the haplotype. And one of the first things that you might notice when you look at this figure is that the red portion of the pie chart is much, much bigger in cases than it is in controls, right? And so this means, and so when you run the statistics on this, you get something called a p-value. And this means that in cases, this particular MHC haplotype is significantly overrepresented in cases. So this was kind of an interesting finding for us, right? And this suggests that these MHC genes, which are important in the immune system, may be playing a role in the development of Addison's disease in Portuguese water dogs. Now, one of the other things that we have to do as geneticists is we have to ask ourselves the question, even though this is statistically significant, how much risk is it actually conferring in the development of Addison's disease? And so you calculate something called a relative risk. And this came up in that previous figure, right, where we showed that a previous group, showed that Portuguese water dogs were 29-fold increased risk of developing Addison's disease. And so what we found here when we actually look at the relative risk of this haplotype, it's actually pretty small. So the relative risk, if you have this particular haplotype, let's just call it the red haplotype, it gives you a 1.6 on about increased risk of developing Addison's disease. So that's not a lot, right? It means that if an average Portuguese water dog, has X likelihood, it's just a little bit greater likelihood if you have this particular MHC haplotype. So while it's important, it's probably playing a role. It's not a major driver of whether or not a Portuguese water dog develops the disease. For those of you guys who are curious, we name these haplotypes. They have these funky names called 00101, 00101, 00201, and that's the DRB1, DQA, and DQB genes. Naming MHC genes as a whole kind of field unto itself. But the bottom line is that this red haplotype, which is technically this 101, 101, 201, confers a small but significant risk in the development of Addison's disease in Portuguese water dogs. Now, another thing which I just kind of want to point out, which is really interesting, which was really interesting to us, is we compared whether or not Portuguese water dogs and standard poodles share MHC haplotypes. And I'm going to sort of ask you guys not to pay too much attention to the figure here on the right, but take a look at the colored bar chart on the left. So if you remember this red bar, this red pie, the red sort of slice in the pie chart on the previous figure was in Portuguese water dogs, the risk allele. You can see standard poodles do not even have that risk haplotype at all. So this is just kind of and overall, the MHC alleles that Portuguese water dogs have are really, really not shared at all with standard poodles. We think that's kind of interesting. We don't exactly know what to make of it. There is a risk haplotype in standard poodles, but it's very, very different than it is in Portuguese water dogs. It's a little unclear that from us as geneticists what to make of that, but that's just sort of an interesting factoid here. Okay, so I'm going to move on and show you guys this next figure, which is the result of the genome-wide association study that we performed. And let me sort of walk you through this figure. I mentioned earlier how we generated this data. This is generated from imputed low-pass whole genome sequencing data. And what you see here is you see all of the dog chromosomes. You see chromosomes 1 through 38, plus the X chromosome at the end. Those are represented on the X-axis. And the Y-axis represents essentially a level of statistical significance. And each dot that you're seeing in this figure is essentially a statistical test for a single SNP or marker in the genome to try to evaluate whether or not it's associated with Addison's disease. And the way we kind of set the bar here is we set the bar for statistical significance at being this blue line here. And the first thing that you notice when you look at this, which is to some extent a disappointing but not unexpected result, is that there is no marker in the genome that statistically separate, no single marker, I should say, that statistically separates affected Portuguese water dogs from unaffected Portuguese water dogs. Now, we would have loved to do this GWAS and find one enormous peak that points us to a single region in the genome, but the reality is that that's just not the case. And so this genome-wide association study essentially rules out, if you remember back to that original figure I showed you, that the cause of Addison's disease in Portuguese water dogs is like a single genetic variant. This effectively rules that out. And it means that understanding Addison's disease in Portuguese water dogs, it's either going to be a polygenic disease, meaning many genes associated with the disease, or to some extent, the predisposition to developing Portuguese water dogs is largely fixed within the breed, and we need to try to understand what that is. I know I'm going a little bit long here, but I'm going to kind of just quickly show you we found something kind of similar in standard poodles, except in standard poodles, we actually did find this peak that you can sort of see. It's chromosome 17. There was a peak in standard poodles that made statistical significance. That same variant did not reach statistical significance in Portuguese water dogs. But when we do a secondary test to evaluate how important that SNP is in the development of the disease, it actually also has a really, really low effect size. So this is just the figure here on the bottom shows us that has a really, really low effect size. So what are the takeaways here from this that I want you guys to remember? For both poodles and Portuguese water dogs, there are some but few genetic differences between affected and unaffected dogs. There are differences in these MHC genes that separate affected from unaffected Portuguese water dogs, but they don't really drive a lot of the risk. Similarly, there's one locus on chromosome 17 that separates standard poodles from unaffected standard poodles, but both of them have very, very small effect on the propensity to develop Addison's disease. And so when we think about there are some kind of blind spots in this that we're trying to close with some ongoing studies. So things that are called structural variants, and there's always regions of the genome that are very, very hard to genotype. But I think we're at a very, very low likelihood of finding a major genetic effect through this through when we ultimately do explore these regions. And so again, this sort of gets back to what I was talking about earlier. You sort of see how grayed out the notion that Addison's disease is a Mendelian disease, and we really need to start thinking about how do we interrogate these polygenic diseases, or trying to understand to what degree the disease could be fixed within the breed. And I've already talked to you guys about this earlier. When we think about map, we sort of then moved on from this initial genome-wide association study to try to understand to what extent this disease could be fixed within a breed. And this represents a lot of the ongoing work that we have. And so there's different ways in which we have of mapping fixed disease predisposition. One way that we have to do it is something called runs of homozygosity. So what happens is when dogs are inbred over time, their alleles tend to become homozygous, meaning there's less variability in those alleles. We can use various computational techniques to try to identify long regions of the genome where there's lots of homozygous alleles, and to try to find regions that are under selection. And then we can also, I talked about this earlier, do things that are called like a cross-breed genome-wide association study. And I'm going to show you some of these things that we're actively working on now. So in Portuguese water dogs, when you look at runs of homozygosity across the genome, this is using the same data set that we used before. You sort of see a similar like plot that I've showed you before, where the height of the peak represents the extent to which it's fixed, and it's sort of all scaled on a scale of 0 to 100, where 100 would mean the entire all Portuguese water dogs are fixed for some of the alleles there. And you can see we have a number of different peaks within this genome, which represent essentially regions of selection within Portuguese water dogs. And so to ask ourselves the question, well, could any of these regions under selection be important in the development of Addison's disease? One of the first things that we did is we asked ourselves, well, if Portuguese water dogs and standard poodles have Addison's disease for the same reason, we would want to run the same analysis in Portuguese water dogs and standard poodles and ask ourselves the question, what regions are being selected for in both breeds? And when you do that analysis, you find 4 regions of the genome on chromosome 8, 10, 13, and the X chromosome that are highly under selection in both breeds. And one of the things that we're working on right now is trying to understand, are there any genes within these regions that we think are going to be highly associated with Addison's disease? And if we do find anything that looks interesting, if there's any variants that could be damaging to proteins, then we would ask ourselves the questions, okay, in breeds with a low risk of Addison's disease, do they have a low frequency of some of these variants? So it becomes a much, much harder picture to stitch together. The other couple findings that I want to talk to you guys about is from the study on spaying and neutering your dog. And so one of the kind of most interesting findings that came out of that environmental risk survey is that far and away the factor that was most predisposed either a Portuguese water dog or a standard poodle to developing Addison's disease is whether or not they were spayed or neutered. And so neutered dogs, so this is for males, had about a 6-fold increased risk of developing Addison's disease, and spayed females had about a 2.5-fold increased risk of developing Addison's disease. And so what does this mean from a practical perspective? It doesn't necessarily mean don't spay or neuter your animals, but it could mean that some of the hormones that are produced by the ovaries, by the testes, are playing a role in the development of the disease. And especially because many of those sex hormones can impact the adrenal gland and its function. And so we can now take this information on whether or not dogs are spayed or neutered. We can overlay it with the genetic information once we have enough once we have enough dogs in our study population to try to ask ourselves the question, if we just focused on the neutered population, if we just focused on the spayed population, can we improve the likelihood of us finding genes that are associated with Addison's disease? So what are our next steps here? So We are in the midst of trying to analyze these kind of shared runs of homozygosity regions to try to understand damaging variants that are shared between poodles and Portuguese water dogs. We're also in the midst of working on some of these cross-breed genome-wide association studies. This is a bit of a can of worms, as I mentioned, because when you start looking across breed, there's lots of differences across breeds beyond just Addison's disease. We also have plans to sort of use different and even newer, very cutting edge genetic techniques to try to, they're called long read sequencing to evaluate many of these structural variants. And then finally, to try to integrate some of the spay and neuter data with our genetic analyses. And I already mentioned this is a challenging thing to do, but I'm kind of we're in the process of working on it, right? And this is just a very, very preliminary example to show you guys what happens when you do these crossbreed GWASs. So this is a quick study that we did using 31 Portuguese water dogs versus about 3,000 other dogs that we have high coverage whole genome sequencing on. You see tons and tons of peaks in this plot. So many different regions of the genome highly separate Portuguese water dogs from the other dogs within a broad population of breeds that we have, 3,000 other dogs. But one interesting hit, for example, this region on chromosome 7, which is almost unique to Portuguese water dogs, is a gene that's associated with immune cell function. It's not a gene that's in standard poodles, but could it be relevant to Portuguese water dogs? We don't know. Every Portuguese water dog is variable, has this particular variant, but maybe it's one of the things that's predisposing them. So this is very preliminary. It's stuff that we're in the midst of working on as we think about perfecting some of these crossbreed genome-wide association studies. So just as I finish up here, and I realize I've gone way over what I said I was going to go, but we'll still have plenty of time for questions. I'm not in any particular rush. I really just want to acknowledge everyone who's been helpful for this study, which is a ton of you guys who are on the Zoom call, in particular the Portuguese Water Dog Foundation, who funded this study through the AKC. I've also had lots of help from the club, the Portuguese Water Dog Club of America, in recruiting samples and just kind of conversations and allowing me to come to the show and collect samples. It's also funded in part, as you mentioned, the Poodle Club of America, and the NIH has given me some money to study this. And then really all of the lab members who've worked on this. So many of you guys may have spoken with Lizzie Grief. She's actually finished her PhD and moved on to do a postdoc at the University of Pennsylvania. She did a huge amount of this work here that went into this presentation. And her primary advisor, who's a great friend and collaborator of mine, a geneticist at the University of Georgia, Leanne Clark, who really has been an instrumental collaborator in sort of working on this data. And Leanne and I are kind of continuing to work together to sort through what could be going on here. So all of that said, I'm going to kind of stop the share of the presentation, and I will open the floor for questions. And I guess just please use the raise hand function, or I'll also open my chat window in case anyone wants to put it in the chat. So thank you guys all for listening. Any questions? You gotta be.
Well, I want to thank you for this presentation, and I don't know if this question is too premature, but for like a male Portuguese water dog, is there an estimated time when they should be neutered?
You know, this study really only looked at whether or not dogs were spayed, whether or not spaying or neutering had an effect. But, I think generally as veterinarians, we sort of encourage people to wait until their dogs are at least full grown before spaying or neutering them, because those hormones can be really important in just general organ, bone health development. So, 18 months to 2 years is the general recommendation. But whether or not that has, how that relates to Addison's disease, I can't necessarily say, but that would be what I would suggest.
Okay, thank you.
Yeah. Claire, hey, good to see you.
Yeah, hi. Thank you so much for a wonderful talk. And I have a question about the
Yeah,
imputation process.
sure, yeah.
Um, how Do you think it would make, and imputation's really controversial in a way sometimes. Do you think it would make a difference to do this study with more dogs with that high coverage? I know that's a lot more expensive, but coming from the Portuguese Water Dog Foundation, like, is that an area you'd like to expand as well? Because I noticed it wasn't in your future comment.
Yeah. So, it's a good question. I actually don't know that, like, I think these imputation is super controversial. I think, when we first started doing it, um, it was like a little bit more challenging, but kind of the way that you predict the genomes has actually become much more accurate. And one of the things that we do when we do imputation, so as you sort of know, you have like a target panel, which is essentially the low coverage genomes, and you have a reference panel, which is the high coverage genomes. So with 31 Portuguese water dogs, which was the number that we have in our reference panel, probably represents the vast amount of diversity across the breed. And we actually picked those 31 Portuguese water dogs specifically to represent the diversity of the breed. So we can sort of look at the dogs that we have on something called, as you know, like called a principal components plot, and make sure that we're capturing as much of the diversity of the breed as we can many times over. And so when you impute, you're essentially predicting those haplotypes that are in the reference panel. And we've used genotype imputation across many, many different traits and other diseases that we study, or that other of my collaborators study, and found it to be really pretty good. And when you compare the accuracy, if you were to sort of take dogs that you have both high coverage on and imputed low pass data and look at the concordance, it's probably like 98 to 99% concordant. And so I don't know that, to answer your question directly, I don't know that adding more Portuguese water dogs to the reference panel would really increase our ability to have more accurate data. I think if we had 5 Portuguese water dogs in the reference panel, I think our output, it would be complete garbage. But given, how we thought about this, I think it's probably pretty good. And when you sort of do high coverage data on like so many dogs, it becomes a huge date. It becomes a big like data challenge. But and anyway, but we do have like our reference panel. We have high coverage data on over 3,000 dogs, but it's just across lots of breeds. So that's how I would think about it. Well, but thank you. That's a great question.
Yeah, I mean, they all go back to two kennels anyway, so they're probably all related.
That's real. And I think that was, that's definitely one of the challenges that we faced with this study is that, even though we collect, almost 500 dogs, to have to eliminate, 4/5 of them because everyone's so related, it makes it challenging. And we're one of the things that we're working on right now is we're working with a group from the UK to try to see if we can get a bunch more dogs that are unrelated to the primarily like US based population to try to improve the number of dogs we can have in the study. But I think it's really I think it's unlikely even it's unlikely that adding a ton more dogs to our GWAS is going to give us, I think, a different result. And I think for us focusing more on these like fixed alleles is really where I think we need to go. And I know as like a breeding community, it's not the result you really want to hear, because you want to know, well, what's the genetic test I can do to sort of remove this scourge from the breed? But the answer really may not be as easy as we thought. And a good example, right, that you can think of is I don't mean this at all in like a disparaging way to anyone who loves bulldogs, but bulldogs, you could make an argument that they are a walking disease, right? Because they have problems with their skin and they have problems with their ability to breathe and they have problems with delivery. And this affects almost every bulldog, right? And so that predisposition to developing those diseases is part and parcel with the way a bulldog looks, right? And there have been lots of people who wrote about this kind of thing. And I think as we start to explore many of these diseases, I don't necessarily think it just relates to Addison's disease, we'll probably start to find that much of these disease predisposition, and maybe not all of it, maybe just some of it, is going to wind up being a little bit more fixed within a breed. And it becomes difficult to think about how do you get rid of that disease without sort of compromising what is it to be a Portuguese water dog or a poodle or that kind of thing. And so these are some of the things that we're working toward, but it's sort of too early because doing these crossbreed GWASs and sort of mapping fixed traits, I think, is something that geneticists are sort of working through as we sort of think about how we handle and how we do things like crossbreed GWASs effectively and things like that. So
I don't mean to dominate, but I think the frustrating part from a breeder perspective is that we do know that certain lines do produce more Addisons and we see it. And
yeah.
so we try to breed away from them. So like intuitively, there does seem to be a inherited component because we sell them to different environments and things like that. So
Totally. And I think you're right. So I'm not going to I'm certainly not going to make any claim that like 100% of the disease predisposition is fixed because we don't know that. But could 50% of it be fixed? Could 70% of it be fixed? And there's these small genetic differences, and identifying these small genetic differences could be really hard to suss out on a GWAS where you only have 100 dogs, right? And so it becomes like, so you wind up having to kind of continually collect more diverse dogs, and that's something that just takes time. So I think you're right. Like, I think it would be impossible. I think it would be really, I'm not at all trying to make the claim that 100% of the predisposition is fixed,
yeah.
because I think that's not consistent with what you see. And I think that's important to say.
But you can see how my imputation question fits in. Like 30 makes sense with central limit theorem and everything like that. But if you're looking for rare event detection and you're normalizing, you might be missing things. But I agree, it's super expensive. There are other avenues you can potentially evaluate, but
Totally. And I think you're right. I mean, I think to the extent that you're trying to map super rare things, it can be challenging with imputation, but to look for like a major risk locus, we 100% like would have found that with 31 diverse Portuguese water dogs. And I mean, another approach you could take, you could just use a snip chip, right? And sort of use a snip chip for more dogs. The problem with the snip chip is that the resolution is not nearly as good as we get with imputation. So, it's a work in progress. Any other questions from the crew?
Yes.
Yeah, go for it. Thank you, Elsa.
Uh,
Yeah.
how was the degree of relatedness defined?
Yeah, great question. So for all the dogs we included in the study, we basically only included them if they were unrelated at
Yeah.
the grandparent level. So we sort of looked at that. And then the other thing which you do when you do these genome-wide association studies, even if you sort of look at the pedigree, there can sometimes be what we call cryptic relatedness that can be difficult to figure out based upon the pedigree. And so we can evaluate based upon other genetic tests how related some of these dogs are using a metric called identical by descent. And then we can actually also correct our like association studies based upon some of this cryptic relatedness. So there's a lot of effort we put into to sort of minimizing the effects of relatedness, but starting with the pedigrees is like our first pass. Great question. Okay, I'm sorry, I'm reading a question here. So since it appears that Addison's is not simple, and since PWDs are so homogeneous, do you see a breeding strategy that could help mitigate? Seems like there's just not enough genetics diversity. So it's a good question that you have here that I'm sort of not going to attempt to answer without a better understanding of what's really going on. Because I think until we know that, it's just like impossible to say like what the right breeding strategy is. Like, I think like I think what you guys are doing, which is essentially breeding away from the lines where you see a high propensity, is probably as Claire was mentioning, is probably as good as you can do at this point. But there's no great answer that I have at this point. I wish I had come out of this 3 or 4 year study with here's the genetic test and you can breed all away from this, but that would have been too good to be true. It would have made my life easier, but also a lot less interesting, I guess is sort of what I would say. But I wish I had a simpler answer for you guys, would also be the simpler. But I mean, just rest assured that we've been really, really thorough in at least figuring out that that's not what's going on here.
Steve, I have a question.
Yeah, go for it.
Carol, is there anything more that we can do as a club to assist you at this point? Or are you in the weeds where our participation is no longer that important?
It's a great question. I think for the moment, I would say a couple things. So first, Cheryl, you were the one who kind of made the contact with the UK group. And so we have sort of a similar effort going on with poodles from the UK, and we're actually able to get the UK people, the poodles and the Portuguese water dogs, to kind of coordinate work together. And that's been like super helpful, and I'm hoping that will be fruitful. To the extent that, we've sort of been down this path and it's been a little difficult to try to find contacts outside of the UK. I'm not sure if there are major populations of Portuguese water dogs outside the US, UK, and Canada, but to the extent that folks have contacts, outside of those countries that have significant breeding populations, that would be for sure helpful. And sorry, there was something else I was going to say as far as things that, oh yeah, and then I think the last thing I would say is, I would say we can never have too many affected dogs. So we have a huge population of Portuguese water dogs. So to the extent that you guys hear, of other dogs that have become affected, please have them reach out to me. We would love to enroll them in the study. And they can email me, you guys, my email address. We also have a separate email set up for the study. It's pwdstudy@umn.edu, where people can just kind of email me directly. But that would also be another thing to do, would be just, we don't really need more unaffected dogs because we have so many at this point. But to the extent that you guys have more affected dogs, that would be fantastic. And the website is still up to promote the study, and we're in the midst of kind of trying to suss out a lot of this crossbreed fixation stuff to kind of figure out where to go. So,
Steve, do you monitor the care group?
Yeah, so we do. Yeah, so we definitely monitor and work with the care group, but. And I think I know Lori Basher pretty well, and we'll occasionally kind of make posts to that website for sure.
Good.
Yeah.
As far as where are the Portuguese water dogs in Europe, the main concentration, interestingly, not necessarily Portugal, where you'd think, it's Finland, Sweden, Norway, and
Okay. Yeah. Yeah, and we have not really, I think we probably have a couple from the Nordic countries. I have to sort of look at our population, but we don't have a ton from there. And so I think to the extent that, people have contacts there, it would be super helpful. They're also obviously perfect English speakers. The way that it sort of has worked for us is for both PWs and poodles, we sort of have one contact, and that contact is someone who's willing to kind of donate their time and be the recipient of cheek swabs. And then once they have enough cheek swabs, they'll send them over to us in the United States en masse. And that sort of just saves on shipping costs, and people can mostly mail them locally to their in-country contact, and then we pay for the shipping to sort of send over the cheek swabs to us, and they can also help with pedigrees and things. So that would definitely be a boon to the study to the extent that we can get some more Nordic dogs enrolled.
Super.
Yeah. Anyone else? Okay. Well, if there's any other questions that you guys have, feel free to shoot me an email. I'm really open to feedback. I love feedback. Like if this weren't for you guys being involved and interested in the study, I'd have nothing to do. And so, I really appreciate all your help and not only like, in terms of getting samples, but obviously like financial support in funding this study. And then kind of continuing to kind of work with us as we move forward and try to figure this out. I think never in a million years did I think when I kind of started my PhD looking at the genetics of standard poodles that I'd still be working on this 10 years later, more because I thought it would be a much simpler problem to solve. But I think this is just kind of part and parcel for where we are and representative of like where we are as like a dog genetics community and trying to kind of understand how breeds and diseases really fit together, beyond once we get beyond kind of like simple Mendelian diseases, which are a lot easier to solve. So yeah, so thank you guys for coming and listening. And Janet, I'll be in touch with you and figure out how we can kind of get you this recording. And yeah, thank you guys for showing up. I really appreciate it. Okay.
And thanks for all your work with this and your update. I know
Absolutely.
everybody really appreciates it.
Absolutely. Thank you, Janet. Thanks for everyone for your help. And we'll be in touch. We'll stay tuned for more. Okay.
Thanks, Steve.
Thanks,
Thank you
guys.
very much.
Bye-bye.